Hi everyone. So far we've been discussing what a microscope is and why we make use of them. The basic idea is that cells are extremely important for us to understand. And the first step in understanding anything is having a good look at it and identifying the kind of structures it possesses. In doing so, we make use of microscopes because they help us visualize these cells that cannot be seen by the naked eye alone. Microscopes were a piece of technological development in biology. And like any other tech device, they're supposed to also have a certain list of specifications that either help us decide that whether this microscope is good or bad. Amongst those specifications, two of the most important specifications are the first one being magnification, which is basically the number of times larger an image is than the real size of that object. And we've already discussed in great length what magnification is and how we can calculate magnification in our previous lessons. In this lesson, we will be focusing on the second most important specification of a microscope, and that specification is called resolution. Resolution, in terms of scientific definition, is the ability to distinguish between two separate points. But I'm sure that doesn't make a lot of sense to you guys. In simple words, it is the ability to see more detail in any image that you're seeing under a microscope. For example, if you compare the two images that you can see in here on your screen, they are the images of the same cell structure viewed under a microscope that had a lower resolution and that had a higher resolution. The microscope with a lower resolution makes this appear as a single point or a single structure, whereas if you look at the same structure under a high resolution microscope, you can appreciate that these are in fact not one single structure, but there is a thin membrane-like line present around both the cells. So it is visible as two separate points under a microscope that has a higher resolution. And in simple terms, a picture with a higher resolution will be clearer versus an image with a low resolution which will appear blur. The idea is that you probably cannot see enough detail on a structure because of either of the two reasons. The first one being that it is not magnified enough. So if you zoom in more on the picture, you would probably see more detail in it. And the second one is because it has a low resolution. And in case the resolution is the problem, no matter how much you magnify it, it will continue to appear more blur, and the structures that were not visible even on a lower magnification will not become more apparent or obvious on a higher magnification. In other words, if increasing the magnification is making the image blurry and more details of the image are not revealed, the problem does not lie in the magnification, but rather the resolution. And increasing the magnification will not be of much help in revealing more detail about that cell or that structure. And just like magnification could be explained through a formula of m is equal to size of image divided by the size of the actual object, similarly, resolution can also be defined as half the wavelength of the radiation that is being used in that microscope. So resolution is going to be equal to half the wavelength of the radiation being used. A little about the wavelength and radiation that is used. In a light microscope, we make use of visible light, which is a small part of the electromagnetic spectrum. An electron beam is a little similar to how X-rays behave on the electromagnetic spectrum. But if you observe, each of the different waveforms or radiations have a different wavelength. The idea is that if the wavelength is short, it means that it has a higher frequency and a higher energy. Higher frequency wavelength will be closer and much smaller and much faster. So they'll be moving much faster and be much closer. Whereas if you observe the wave pattern of a wavelength that has a longer wavelength, you can see that it is spread further apart and it has a lower frequency and a lower energy.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
I promise that this has a correlation to what I'm about to explain next. The idea that resolution is equal to half the wavelength means that if an object that is equal to the size of half the wavelength that is being used, it will actually interfere with the projection of that wavelength and will probably block that wave. So for example, light, the smallest wavelength of light or visible light is around 400 nanometers, and half of 400 nanometers is 200 nanometers. So anything that is either 200 nanometers or even 201 nanometers, so anything that is even slightly larger than 200 nanometers will be big enough to block the wave of that radiation. But let's say we talk about, like right now in this diagram, you can see that the mitochondrion shown has a diameter of 1000 nanometer, which is definitely much bigger than 200 nanometers. So it interferes with the light waves and blocks its projection. Whereas if you observe the ribosomes being shown within this diagram, you can observe that these ribosomes are only 25 nanometer, which is much, much, much smaller than a 200 nanometer. And therefore, they do not block the progression of light waves, and light can continue to pass through regardless of the fact that these ribosomes are right there lying in between the progression of that wave. Which is why through a light microscope you can see the mitochondrion, but you cannot see any ribosomes. The idea is that in case of a light microscope, which will have a resolution of 200 nanometers, because half of 400 is equal to 200 nanometers, which means that the maximum resolution a light microscope can have is 200 nanometers. And if you now wonder what does it even mean to have a resolution of 200 nanometers, it means that any object that is closer than 200 nanometer cannot be distinguished as two separate points by the light microscope and would appear as one. So for example, if anything has a size of, let's say, 180 nanometers, that object will not be identified by the light microscope as a separate object, and it would appear as a single object. So remember the picture that I've just shown you on top? That's why you cannot see that membrane-ish line as a separate point, because it is not further away from this surrounding structure by more than 200 nanometers. If it was further away than 200 nanometers, let's say if it was even 200 nanometers or 201 nanometer, it would have appeared as two separate points. But now let us come to the electron microscope, which is known to have a resolution of 0.5 nanometers. So there were two types of electron microscope, transmission and scanning. The transmission microscope is known to have a resolution of as low as 0.5 nanometers. That means it can differentiate any point as long as it is away from each other at a distance of 0.5 nanometers. And that's because the electron beam has a much shorter wavelength, which means it has a much higher frequency. That makes it look something like this. And in doing so, for example, now ribosomes, which were too small to be picked up and be blocking the light waves passing through, are now large enough and at least meet half the wavelength of an electron beam, and therefore they are able to block that electron beam from progressing forward. Which is why under an electron microscope you can see ribosomes, whereas under a light microscope you could never see the ribosomes. So the point being, if an object is more than or equal to half the wavelength of the radiation being used, it will end up interfering with its movement and be visible as a separate object. If it is smaller than that, it will not interfere with the radiation and cannot be distinguished as a separate object or a point under that microscope. Now, going back to the image I showed you, the first image is taken from a light microscope and the second image is taken by an electron microscope. So an electron microscope has a resolution of 0.5 nanometers. That means any objects even as close as 0.5 nanometers will be identified by this electron microscope as a separate structure. Whereas the light microscope has a resolution of only 200 nanometers, which means any object that are closer to each other than 200 nanometers will not be identified as separate objects. So imagine a distance of only 0.5 nanometers and still being able to identify them as separate versus having a resolution of 200 nanometers and not being able to identify any object that has a distance or has a size of smaller than 200 nanometers. Which is why this membrane is visible as a separate point and a separate structure under this picture, which is an electron micrograph, and is not visible as a separate point in a light micrograph. Which brings me to the last point on this lesson, which is that as long as you are magnifying up to the limit of resolution, it will reveal more detail. But any further magnification than that will not increase any detail, but instead will increase the blurring of that image. Which is why if you go back to this image, and you notice that upon increasing the magnification, the details on this image are not increasing. And in fact, the picture starts to look more blurry. And that's because within this image, we are magnifying this image beyond its maximum useful magnification, which corresponds to the best resolution. So for a light microscope, the best resolution a light microscope can have is 200 nanometers. And the maximum useful magnification for a light microscope corresponds to 1500 times. Any magnification beyond 1500 times will only result in blurring of the image and will not reveal more detail. In contrast to this, an electron microscope has a maximum resolution of 0.5 nanometers, and the maximum useful magnification that corresponds to 0.5 nanometer resolution is 500,000 times. So you can actually magnify an image 500,000 times and it will continue to reveal more and more detail of a structure that you're viewing under a microscope, beyond which it will start to blur. Which is why when you looked at a regular cell under a microscope, you could appreciate more structures than a light microscope. But let's say initially, when you looked at an animal cell under an electron microscope from a light microscope, you could appreciate a lot of extra structures. But for example, like ribosomes were never visible under a light microscope, but they are visible under an electron microscope. But initially, at a lower magnification, they appeared as tiny dots. But as you went along and decided to magnify the image even more and more details were revealed about that ribosomes, and we learned and observed that ribosomes are actually not tiny dots. They actually have a smaller subunit and a larger subunit. So the more we zoomed in and the more we magnified that image, the more detail we could see and appreciate. Similarly, mitochondria was just visible as tiny little sausage structure under a light microscope. But under the electron microscope, even without magnifying it too much, we could observe that it was a double membrane structure. And you will learn more about these structures when we're doing cell structure. But we basically learned far more, or we basically could see far more details within the structure that was already visible under the light microscope. But we could never have assumed that it is such a complicated and intricate structure inside that tiny little structure that only appears as a sausage-shaped structure under a light microscope. And as we went on to further magnify it, more and more detail continued to reveal itself, because we kept magnifying until we reached the maximum useful magnification. And after increasing the size of the image by 500 times, after that, if you tried increasing it, let's say you tried increasing it by 501,000 times. So if you ended up increasing the magnification beyond that limit, then the image started to get blurry and it started to not reveal more detail. But to summarize this lesson in a nutshell, resolution is the ability to distinguish between two separate points and is equal to half the wavelength of the radiation that is being used. Higher the resolution, clearer will be the picture, and lower the resolution, the less clear or blurry that picture will appear. And on that note, let's conclude this lesson.
Hey there! If you found this A-level biology lesson helpful, why don't you head over to altacademy.org and sign up for our full A-level biology course. In addition to biology, you could sign up for physics, chemistry, math, or 4 other A-level subjects. Thousands of students have already taken the leap. What are you waiting for? See you there.